Good morning. Today I'm going to talk about DNA and genetics. This is Mrs. Spencer, and this is the third session Biology EOC review video. So we're going to go over DNA and genetics in this particular session, and I do thank you for joining me on this review session. Alright, so we're going to start by talking about DNA, and just remember that DNA is a organic compound. It's one of the 4 main organic compounds found in living things, and it is the basis of all of our genetic input. So it is kind of like our blueprints, and that is what makes up all living things, no matter as complex as you are from a human to something very simple like a bacterium. Quick review, DNA is found inside the chromosomes in the cell, which is also called chromatin. This is the little squiggly, spaghetti-looking stuff that's inside the nucleus of most cells. In bacteria, it's just a single strand of chromatin. And it is made up of nucleotides. That's the building block of DNA. Those are the smaller compounds that make up DNA, and DNA is considered a double helix, which means that it is actually 2 strands of nucleotides that are wrapped around one another. Let's take a little bit of a closer look at a nucleotide. Nucleotides are a 5-carbon sugar, so each nucleotide has a 5-carbon sugar, and it's also going to have a base, a nitrogenous base of some kind, and then and then it's also going to have a phosphate group. So a sugar, a base, and a phosphate group makes up one nucleotide. The sugar in DNA is deoxyribose, which is where the D comes from in DNA, deoxyribonucleic acid. So that is the type of sugar, and the bases are either adenine, thymine, cytosine, or guanine. Those are the 4 different bases that can be found in each nucleotide. And it is these different sequences and combinations that make us all unique. Now, when you add a nucleotide to a nucleotide to a nucleotide to a nucleotide, we get DNA. And notice it's 2 strands, so you got 6 total nucleotides in this particular DNA that is drawn here on this slide. 3 on one side and 3 on the other side. And notice that when the bases are pairing up with one another in the DNA, they are complementary, which means that they have a specific base that they will match up with. So adenine always matches with thymine and vice versa, and guanine always matches with cytosine and vice versa. So that is what we call complementary base pairing. Alright, so let's just kind of review a quick check here and just to make sure you understand this complementary base pairing. If we were to have this sequence of bases in a DNA molecule, what would be the matching bases on the other side of the molecule? And they would be the complementary. So the A would match with the T, this T would match with an A, the C would match with the G, C would match with another G, and so on and so forth. So you can see that they are complementary. So that would be one side of the DNA in the black, and then the other side is in here in the red. Alright, now when cells get ready to divide, the new cells have to have copies of the DNA in order to know what to do and what, to build as far as proteins are concerned. So every new cell has to have a copy. So the cells have to replicate or make copies of their DNA prior to cell division. And if you remember from the cell division unit, that's going to occur during interphase, and the S portion, the synthesis portion, is when DNA gets replicated or copied. So the way that DNA does this is that double helix, that 2-stranded DNA molecule, is going to split down the middle. It's going to split in between the nitrogenous bases so that the strands come apart from one another. And then pieces of the nucleotides that are in the cell are going to come in and build another side of nucleotides to each of the broken apart pieces. So you end up with 2 new DNA strands, and the 2 new DNA strands are going to have half of the old strand and then half new. So they're kind of made up of a little bit of both. But in the end, you'll end up with 2 DNA strands that were identical to the original. Okay? So that is called DNA replication. And again, that only happens prior to cell division. Now, the rest of the time, the cell is growing and working, so it has to build proteins. And the proteins are what make up the organism and are what do the jobs. So we have to turn these blueprints into a house, so turn the DNA into the proteins. And this is done through protein synthesis, which can take place at any time in a cell's life cycle, except for when it divides. During division, protein synthesis is not necessarily taking place because the cell is actually dividing. But every other portion of the life cycle, protein synthesis is happening. And this is just how proteins are made from the DNA instructions, because it's almost like those DNA instructions are in a different language that the cell can't understand. So we have to have some way of converting the DNA into a language that's understandable and actually being able to build the proteins. So there's 2 main steps. There's transcription and translation. Those are the 2 main steps to the building of proteins. In transcription, this is the first step. The DNA is going to unwind, much like it did for replication. It's going to unwind and unzip down the middle, okay? And we're going to get a new strand of nucleotides to build alongside one of the DNA strands that was split apart. But the difference here is that this time, when the new strand is being built, it's being built as messenger RNA. And nothing really is different here except the sugar. Okay, RNA has a different sugar, which is ribose, instead of deoxyribose. So there is a new sugar in the nucleotides. And we have a different base. Instead of thymine, it's uracil. So adenine will match with uracil instead of thymine in this process, okay? But the G's and the C's still match up. And if there was a thymine on DNA, it's still going to match with an adenine on mRNA, on the messenger RNA. It's just when the adenine is on DNA, it can only match with the uracil. There's no thymine in RNA, okay? And so the messenger RNA is going to build based upon the DNA. Basically, it's like it's getting that code with a little bit of a translation happening in that change of the thymine to uracil. Okay, but notice it's still a complementary pairing. So the A on DNA matches with a U on RNA. The C on DNA matches with the G. Okay, so you're still complementing here. Once the messenger RNA strand is built, it's going to separate itself from the DNA and leave the nucleus and go to a ribosome in the cytoplasm. Because ribosomes, if you remember, are the actual things that build the proteins. So that's what needs to get this code. So the messenger RNA leaves the nucleus and goes to a ribosome. So that's the transcription part, the actual getting of the code onto the RNA and taking it to a ribosome. The second step is called translation. Translation is at the ribosome, and this is where the ribosome reads the code on the messenger RNA and translates it into the building blocks of proteins, which are amino acids. Every 3 nitrogenous bases on the RNA equates to 1 amino acid or building block of a protein, okay? And each sequence has a specific amino acid that it codes for. And the messenger RNA, the ribosome is going to read and understand what those are. So those amino acids are brought over to the ribosome when needed, when they're supposed to come in place, and they're attached together by the ribosome until the messenger RNA gets to a code that basically says to stop. And then that's when the protein is finished and is released to do its job. And that is called translation. Okay. Every 3 bases is also called a codon, because it's basically the code for one amino acid. All right? And notice there are start codes and there are stop codes, much like a capital letter indicates the start of a sentence and a period indicates the stop of the sentence. So the same thing's going to be true here in these codes when building proteins. We're going to have starts and stops so that the ribosome knows when to start building the protein and when that protein is done being built. Okay. I want to go back one slide just to kind of review a little bit more about that RNA. Remember that RNA is another type of nucleic acid, but it's only a single strand. It is built up of nucleotides just like DNA, but the difference is that the bases are not going to have thymine. Adenine, uracil, guanine, and cytosine are in the RNAs. Okay, and then again also the sugar is ribose. So here I just wanted to show you how when the DNA has this sequence in the black of bases, the messenger RNA that's going to come up and complement that when the protein synthesis is taking place is going to complement much like it did with DNA, except that anytime where a T would have normally gone to complement, it's now going to be a U. Okay? So there's no thymines in RNA. All right. Now, sometimes mistakes do happen, and sometimes that causes a change in the genetic code. So this tends to happen when the messenger RNA is being built. For some reason it misreads the DNA and we get some mistakes and it changes the actual code that was on the DNA, and this is called a mutation. So there are some different kinds of mutations we are showing here. A deletion is where one of the nitrogenous bases is left off. Okay, so that shifts all of these codons, because every 3 is supposed to code for a specific amino acid, as you can see here. And what has happened is we've got, in this deletion, the first 2 are still okay, but because that A in that 3rd codon has been deleted, it shifts the codon down, so now the codon reads UCC, which is a completely different amino acid than what it should have been in that particular spot. So that's going to change the protein and cause the protein to not work properly, and that can then of course lead to serious problems in the organism. Insertion is kind of the same thing as far as the shifting, only this time we add an extra amino acid. An extra nitrogenous base. So again here, this time the first 3 amino acids are correct, but there's an extra U, an extra uracil that got added in for whatever reason when the messenger RNA was building it, accidentally put an extra base in, and this shifts everything over, and so now we get, again, it's supposed to be proline after the isolene, the pro, but we have a serine instead, the SER. And so again, that's going to change that protein and cause it not to work. And then substitution is where instead of one nitrogenous base, it's substituted with a different one. So in this case, the G, the guanine, was substituted with a C accidentally. And again, this changes the amino acid, and that changes the protein in essence. So all of these, in these cases, because the amino acids are changed, the protein also is changed, which causes some major problems in the individual. Now, occasionally, a change can occur, and it still codes for the same amino acid, because there are multiple codes for some of the amino acids. So sometimes a change can happen and the amino acid still remains the same, so the protein doesn't get changed at all, and so there's almost as if there was never a problem. So we do have mutations that sometimes happen that, you know, are not a big deal. but unfortunately, a lot of times they are. All right, so now let's talk a little bit about how the DNA equates to what we see and what the traits are of an organism. So inheritance, basically your genetic inheritance. So proteins, again, are made from the DNA code, and each section of the DNA code are known as genes. So, you know, in this particular case, here's the nitrogenous base sequence. And you can see that all of these blue bases equate to one gene, which happens to represent the trait of pea plant color, characteristic or the trait of pea plant color, which can either be yellow or green. Those are called alleles. Alleles are the different forms of the trait. So in the case of pea plant color, the pea plant can be yellow or can be green. Okay. The yellow nitrogenous bases over here on the DNA represent a separate gene, gene number 2, which happens to be representative of height, the trait of height, which can have either tall or short. Okay, so that's, in this case, all that is on this particular chromosome. Most chromosomes, especially in complex organisms like ourselves, are going to hold hundreds to thousands of different genes on one chromosome, okay? And the DNA that's in those chromosomes is, you know, massive. So, but basically what I'm trying to show here is a simplistic view of it, showing you that, you know, a section, a whole, you know, the DNA has thousands of different bases, and different sections of those represent different genes, which represent different traits, which can be shown up in different forms. Okay? All right. So let's talk a little bit about Mendel. Gregor Mendel, was a monk who did some experimentation with pea plants. And he is known as the father of genetics because he did a lot of probability stuff, math stuff with the transfer of different traits in pea plants from one generation to the next. And from his work, we were able to get a lot of basics in the regards of how things get passed from parent to offspring. One of the biggest things that he gave us is the term dominant and recessive. In most cases, traits have a dominant form and a recessive form. The dominant form is the form that tends to always show, even if it's matched with another recessive. It kind of overpowers the recessive. The recessive of a form of a trait is going to be less likely to show, except for when you get a recessive matched with the recessive. Okay. When we write this out, if we were to actually, like for instance, write out tall versus small or short in pea plants, tall happens to be the dominant form. It happens to be the one that kind of overpowers. So we would represent that with a capital T because of it being the dominant. Whereas short, since it's the recessive, that would be represented with a lowercase letter, and we still use the T just because it helps us remember that we're talking about the same trait. So the capital T represents tall, the lowercase t represents short, and this tells us that tall is dominant, recessive is short because of it being the lowercase letter. Okay, and these are the two alleles or forms of height in a pea plant. Okay, and the way we know that tall is dominant versus the short is based on the inheritance patterns that are shown from parent to offspring. And so we'll go over a few of these. So we get one set of chromosomes from mom and one set of chromosomes from dad. So we actually end up with 2 copies of each gene. Okay, so you have one copy of the height gene from mom and one copy of the height gene from dad. So in the form of color of pea seeds, we can have a yellow or a green. So you can either get 2 yellows one from mom and one from dad, both yellow. You can get a yellow from one of your parents and a green from another parent, so you end up having one of each, or you can have two greens. Okay, and again, notice I've used capital and lowercase y's here, and that is because yellow is the dominant trait, the dominant allele, and green is the recessive, so that's the lowercase y. Okay, so when you have, if you happen to get two of the yellows, one yellow from mom, one yellow from dad, then you're going to automatically have yellow seeds. Okay, however, if you get one yellow from mom and one green from dad, or a yellow from dad and a green from mom, one of each basically, even though you have the green allele there, the yellow overpowers it, so you still will have yellow seeds. But you can still pass that green color on to an offspring. So you could have offspring with green, okay, because it's hidden. If you get two green genes, then you have green seeds. So the combination of the actual genes is called the genotype. So whether you have two of the dominant, two of the recessive, or one of each, that's called the genotype. The actual appearance, what you see, is called the phenotype. Pheno for physical, that's what we see. So this is what actually shows up. And you can see that with the combination of the two different ones, even though your genotype says you have one of each, your phenotype looks like you only have yellow because that's what we see. Okay? Thing to remember is that recessive genes, recessive forms of a trait, will only show up, will only be seen when you get two of them. You can only see a recessive when you get two recessives together. Okay? So genotype is this, phenotype is over here what you see. Let's take a look at the Punnett squares. Punnett squares are just a way to show possible offspring genotypes and phenotypes, so you can kind of predict the possible outcomes of offspring. So the way we do this is you set it up with one of your parents on one side and one of the parents at the top. And it doesn't matter which one's where, as long as you have one on one place or one on the other. And then you fill in the boxes. So for this case, we have a parent that is going to have a yellow gene and a green gene, and another parent with a yellow gene and a green gene. So both parents, even though both parents have yellow seeds, they are both hiding a green gene, so they can pass the green on. So what that tells us is we can possibly get a yellow from one parent and a yellow from another to combine, and that's what we're showing in this first box here. And in this case, they would have nothing but yellow genes, so they would have yellow seeds. In this box, we have a green coming down and a yellow coming over. So you have one yellow gene and one green gene. Again, we're going to see the yellow peas, even though the green is hidden, but that's the yellow is going to overpower. In this particular one, we have a yellow coming from parent 1 and a green coming from parent 2, so we get one of each. Again, yellow overpowers, so we see yellow, but the genotype, they're hiding a green. But there is a chance of a green from one parent and a green from the other parent to come together. So they do have the possibility of having offspring with green peas. Okay, it's a small possibility, but it's there. So if I were to ratio this or try to look at the percentages, the genotype percentages would be a 1 to 2 to 1. There would be a 25% chance of getting both dominant, a 25% chance of them having both recessive, and a 50% chance of them having one of each. But the phenotype, it changes a little bit because remember, phenotype's what we see, and those mixed ones will still show yellow. So with the phenotype, there's a 3 to 1, 3 times more likely to get yellow offspring and 1 times likely to get green. Or another way of saying this is 75% chance of having yellow offspring, only 25% chance of having a green offspring. But there is still a chance. Okay, so that's what Punnett squares are for, and that's kind of how we interpret them. They show us the possible offspring. Now, over time, we have determined that there are some exceptions to Mendel's laws. Okay, there are cases where there's codominance, and this means that instead of just one dominant allele, there's 2. And in these cases, both of the alleles show up. And so that happens a lot in like hair color of cats and dogs, chicken feather colors. So what this means is that instead of just having one of the forms to be dominant, you actually have 2 dominant. So in the end, you see both. So for instance, with the chickens, you can have chickens that have both black feathers and white feathers because the 2 colors are co-dominant. So if they get one of each of those genes, instead of one of them overpowering the other one, they both show. And that's called codominance. Co means to share. Multiple alleles means that there's more than 2 forms. What we looked at before was basically, height is short or tall. Pea color, green or yellow, that's just 2 forms. But sometimes we actually have more than that. And then a good example is in the blood type of humans. In blood type of humans, we have 3 alleles. We have A, we can have blood type A. We can have B, which is just blood type B. We also have an O blood type. An O blood type, so that's a 3rd blood type. So there's actually 3 different forms that can show up. And blood type in humans, not only can you have these 3 different alleles, but when the A and the B come together, it's codominant, so they both show up. So that gives a 4th form, really. Okay? Because you can have just A, just B, just O, or you can have both A and B. So that's another exception to Mendel's, like black or white, tall or short, yellow or green. Okay? So those are just a few exceptions to his laws. A couple other exceptions are polygenic. Poly means many. Genic refers to genes. So we do have, especially in more complex organisms such as our cells, we have multiple genes that control a single trait. For instance, eye color is the trait, but there are, I think, 7 or 8 different genes that control eye color. So you can't just do one Punnett square because that would only represent one gene. You would have to do a Punnett square for each of the genes, and then they can all combine in different ways to give different colors. So this is why we can have all different shades of eye color, and why parents can have children with all different shades. Even though they may be both brown-eyed, they can have a blue-eyed son and things like that. It's not a simple dominant recessive with one gene, even though a lot of times people do think that blue eyes is recessive to brown eyes. It kind of is, but it's more than just one gene. There's lots of things that go into what color eyes you have. Same for skin color, and same for height in humans. Height is not a simple short or tall. That would mean we would just have 2 different, heights of humans, and we know that that's not true. So that's because they're controlled by multiple genes all interacting with one another. Sex-linked is another exception to Mendel's laws. Mendel's laws kind of say that everything's inherited independently of one another. And that's true if they're on separate chromosomes. But we do have traits that are actually linked to each other. So like for instance, the trait, as far as whether you're a boy or a girl, your gender is linked to other types of traits. Okay? Remember that boys are going to be have an X chromosome and a Y chromosome. Girls have 2 X's. Okay? And a lot of times when we are talking about a sex-linked trait, we're talking about something that is linked to the X chromosome, which means that it's inherited with the X. So girls will be able to get 2 X's, so they can sometimes hide a trait that's on the X. Whereas boys, since they only get one X, if they have that particular trait, then they're going to always show it because their Y won't do anything because it's completely different. So we're going to look a little bit closer at these sex-linked traits, okay? This first Punnett square up here is showing you basically the probability of a mom and a dad having a boy or a girl offspring. Mom has two X's, dad has an X and a Y. So there's a 50-50 chance, a 50% chance that a girl will happen, and a 50% chance that a boy will happen. So it's 50-50. And it's 50-50 every time. If you have a girl the first time, it's still the same thing. It doesn't take away one of these. You have the same 4 options again the next time, okay? So always 50-50 chance. Whoops, lost my page. Okay. All right. Down here, though, we're showing you baldness. Male pattern baldness happens to be a genetic trait that is on the X chromosome. So, boys out there, if you want to know if you have a chance of becoming bald when you get older, you want to take a look at your mom's dad. Your mom's dad. Okay? All right. And if your mom's dad, so if your grandpa on your mom's side is bald, there's a good chance you will end up being bald. Okay? And that is because it is passed down from your mom. All righty? So you can see here, mom has 2 X's, and mom's got a normal hair gene on one of the X's, and she's got a baldness gene on the other X. Okay? Dad over here has 1 X, and his happens to be normal hair gene, and then the Y. Okay? So now, when the possible offsprings are here, we can get the X from dad and the X from mom here to come together for a girl with 2 normal hair genes. Or we can have this X from dad to come together with the other X from mom to get 1 X with the normal gene and 1 X with the bald gene. But again, it's still a girl with normal who's going to show normal hair, because that normal hair is dominant. It's going to hide the baldness gene. So the girl won't have any baldness. So see how she's able to hide it? Because she's got a second X to overpower. She's got that dominant X that can overpower it. Okay? However, if you take a look when we put the Y's, the boys come from, in order to make a boy, you have to get a Y from your dad. So we have the X from mom coming together with the Y. In this case, we've got normal X gene with a Y, and so this boy will have normal hairline. But if this X right here from mom, the second X with the baldness gene, happens to match up with the Y, then the boy will be bald because the Y can't help you. Okay? So, there's 100% chance that all the girls will show, for phenotype, 100% chance that the girls will be normal hairline. But when we look at the boys, it's a 50-50 chance. Okay? 50% chance that a boy will have normal hair, 50% chance that the boy will have baldness. Okay? And again, it's 50-50 because we're only looking at boys here. All right? If we just said children in general, it doesn't matter if they're a boy or girl, then it's a 25% chance that they'll have a child that will be bald. But if you are trying to specify gender, then you have to look only at that gender's boxes, and in this case, that's just 2, so it's half and half. Okay? And notice that we put the letters are, you know, kind of a superscript, and that's just because we want to show that it is linked to that sex chromosome and the gender as well. So you kind of are looking at both together, the gender and the baldness trait. A few other things that are linked to the X, just for your own interest, if you want to know, there's color blindness is another big one that is linked to the X chromosome, and that's one of the reasons why more boys tend to be colorblind than girls, because again, they only have one X. Boys only have one X, so it's a little easier for that recessive colorblind trait to show up. The girls can hide it. In order for a girl to be colorblind, they have to get two recessives together, and it's a little harder for that to happen. Okay, so that's another one that's on the X chromosome. All right, so that is sex-linked. Okay, and the last thing I want to talk about in this session is basically biotechnology and the alteration of genes and kind of genetics and DNA and stuff, because that is something that is a big hot topic right now, and more is being done with it. And you know, lots of decisions are going to have to be made and are starting to be made as to the ethics and the lines that we should and should not cross when it comes to this stuff. So I want to go over a couple things. Genetic engineering is basically the changing of an organism's DNA. That is what genetic engineering means. It means you are directly changing the organism's DNA. And this can be done in what's known as gene therapy, where you basically take the DNA of an organism out of a cell and you insert the new DNA into that so that the gene will be fixed. Okay? And then there's genetically modified organisms, GMOs. That's a hot acronym that's thrown around a lot. GMOs, you know, you don't want to eat. People are saying you don't want to eat genetically modified crops like oranges, you know, fruits and vegetables and things like that. Anything that's been genetically modified. So a lot of crops are genetically modified. And what has happened is that they've used this gene therapy in a lot of different plants and crops in order to basically put natural pesticides into the crop so that they can, you know, be resistant to pest and fungus and things like that. So, you know, it depends on what you think, and you know, these are things you want to learn more about, so you can have a you know, informed decision when you are deciding whether you agree with it or don't agree with it. You know, one way to think of it with the genetically modified stuff is, would you rather have the orange that was genetically modified to naturally fight the pest by using the DNA of another organism, or would you rather it be sprayed with the chemicals to fight the pest, which are synthetic? That's you know, or do that's a personal preference, but that's just some things to kind of keep in mind. Many people, you know, are not in agreement with changing the actual DNA of an organism, and you know, saying that is crossing a line, and that is completely up to you. Cloning. This is another big hot topic. This is where you actually make an identical copy of an organism. So you're not just altering a specific trait or gene, you're actually copying the individual completely. Okay? So you might have remembered, and you guys might be a little young still for this, but you may have heard of Dolly the sheep, which was a sheep that was completely copied from another sheep. So the thing with cloning you got to keep in mind is that sometimes there's a misunderstanding that if I were to say clone, you know, my dog Peggy, who is 15 years old, that when you finish with the cloning process that you're going to have this 15-year-old dog automatically, and that's not the case. So that you clone to where it will start as a fetus and a baby and grow, and so it's going to have different life experiences and things. So although genetically the clone is identical, actually in the end there's going to be behavioral differences and things like that. Part of a problem with cloning and having identical copies is that you are limiting the variety in that population, in that organism, and so it makes it a little bit more difficult if there were ever to have some major changes in environment and things like that for them to survive, because everything's exactly the same. So variety is actually a good thing in a population, because it allows for you know, some of the population to maybe survive when there is change in the environment because they're not all exactly the same. So there's good things and bad things about cloning. Cloning does naturally occur. Just do know this. We actually have cloning occurring in our bodies when our skin cells replace themselves or heal from a cut. They are making genetic identical copies of cells to replace them. So that is natural cloning. So there is some natural cloning in nature for growth and repair. The question is, should we clone for other reasons? And that's where we start to have the line of ethics and what should we and should not do. Okay? Stem cells. Stem cells are another hot topic. Stem cells are cells that are not differentiated yet. They have not become specific cell types. So for instance, they are just kind of generic, and they can become any type of cell in the body. So they are a hot research tool right now because people are wanting to use stem cells to try to regenerate things like nerves and hearts and stuff that normally will not basically fix itself once it's grown. If it becomes injured, that's it. Like when people have accidents when they're older and they become paralyzed, if we can use stem cells to regenerate those nerves and nerve tissue to replace the damaged nerve tissue, then maybe we could fix or cure their paralysis. Same with people who have heart attacks. Maybe we can replace the damaged part of the heart, and the person won't be likely to have a shorter lifespan because of the heart attack or the injured heart. So that's a big thing with stem cells. The controversy with stem cells is where they come from, because unfortunately the undifferentiated stem cells are early stem cells, so they have to come from things like fetuses, and but not just that. They can come from the umbilical cord, which is not an actual fetus. They can come from other early parts of living things. They don't have to actually come from a baby, so to speak. But that is a hot topic and a point of controversy as to whether or not this should be done as well. Lastly, I want to talk about designer babies. This is something that's becoming more and more of a issue, and this is because we're able to do this genetic engineering up here and alter and change genes and things like that. Now people are talking about, and in some countries are doing, where they are basically choosing babies with specific genes by having the genetics from mom and genetics from dad to come together to create, what they're the genes from mom and dad. So it is part of the mom and dad, but basically choosing gender, for instance, choosing whether they have a boy or a girl, looking to see if there's a genetic disorder, and if there is, not using that particular fertilized egg or gamete sex cell, instead using something else. So, and again, this is another fine line of, are we crossing the line here, and things like that. So these topics are all topics I encourage you to find out more about and really, learn the entire aspects of what they entail, and see both sides of the spectrum, so that way you can make an informed and educated decision on where you stand, because it won't be long before you will probably be voting on issues in the government that will be making laws based on these things. So you really need to understand these pretty well, okay? And that is it for this particular session. And I do thank you guys for joining me, and we will be continuing on with a few other video sessions to prepare for that EOC. And have a wonderful day. Thank you.